MCQs. We started a new series which is about MCQs from Sindh Accdemy Umerkot. In last video we did enzymes few MCQs. Today we'll go with the remaining MCQs. So let's begin. The first question says primary function of Golgi complex. The Golgi complex is simply an organelle in cell, which is very important because this was simply given by a scientist called as Camillo Golgi in 1898. The function of this is to modify packaging of secretion and to transport, like if they made protein, now this would be given to, for suppose ribosome has made protein, right? Because ribosome are called as protein synthesis, protein factory. They have made, now the transportation work would be for the Golgi complex. So right option here is C, which is modification. Why? ATP production, no, this happens in the mitochondria. DNA replication happens in the nucleus. Detoxification, yeah, this happens, like let's say liver, if you have liver, in that we have SCR, which helps in the detoxification of drugs and some structure, like modify them like that. Okay, next question says according to induced fit model, effectiveness is mainly, so we have two models. First one is lock and key model, second one is induced fit model. These are the two models. Like for suppose say lock and key model was given by Emil Fischer, scientist, in 1898, and induced fit model was given by Koshland in 1959. Okay, so this is very important, this lock and key. Even if we see lock and key, we might, like we can easily get lock and key. This means this is a particular key, a particular lock can be unlocked by particular key. So we need, for suppose if this is enzyme and we need a, if this is a substrate for suppose, and we need an enzyme particularly for this shape to be fixed, right? So this was said by Emil Fischer. Okay, lock and key model. But induced fit model was different. They said if this is a, for suppose substrate, and we do not have an enzyme particularly for this shape, for suppose now we have like this kind of shape, but still it could be fixed by changing its position. Now this will change like this, or this could change into this position. So this means they says they can change the enzyme shape, not this shape, but the enzyme shape can be changed. So here B is the right option due to substrate induced conformation change in enzymes. Okay, then this is primary role of cristae in mitochondria. Mitochondria structure is like this. It's a double membrane bound organelle. Why? Because they do follow endosymbiotic theory. This and chloroplast as well, they both have some kind of membrane, ribosome, all of, they can function themselves alone as well, but it's just they need much protein, much other function which are controlled by nucleus. So this is a double membrane bound organelle like this. Inside this we have structured this kind of, this is called as cristae. Cristae are simply curve. Why these are interface area, when surface, wherever you just see some curves in human body, in some, for suppose if we take example of large intestine, small intestine, there we have this kind of structure. So curves are very important. There we have villi like structure, they help in increasing surface area. Same goes for here. So this is outer membrane, this is inner membrane, and the middle region between both is called as intermembrane space, and we do have some ribosome on this. So the question says what is role? So this simply is increase surface area for electron transport chain, etc happens here, so that's why they increase. Let's see, and there is a question, smooth endoplasm transport protein manufactured by RER to which organelle? They say same. For suppose RER, which means protein or synthesis here, now it will be manufactured, transported, synthesized by which organelle? Now these all are the function of Golgi apparatus. Okay, then question comes, enzyme that's act outside the cell, that's called as exoenzymes, which works outside. Feedback inhibition is an example of non-competitive inhibition, right? Make sure you learn this, it's very important in MCQs. Let's go to another, which structure differentiate eukaryotic nuclei and prokaryotic nuclei region. So we have eukaryotic and prokaryotic different structures. So this is simply the right option here is A, presence of nuclear pores, because these are only present in prokaryotes, not in the eukaryotes. Circular DNA, this is property of only prokaryotes. They have circular DNA. Lack of membrane, yeah, prokaryotes. Association with mesosome, these all are a function of prokaryotes, not the eukaryotes. Let's go to another question. This is identify component of holoenzyme. So holoenzyme is kind of different structure. It's simply a polypeptide chain, simply cofactor, collectively called as holoenzyme. Cofactor is simply non-protein part, right? But it consists of polypeptide chain. Then we call them holoenzymes. So they have polypeptide chain, they have RNA, cofactor, DNA. So what's the right option? So the right option here is C. Why? Because as I mentioned, they are polypeptide chain and cofactor. Cofactor is simply a non-protein part, so this is the right option. 43 question says chromosome become visible during cell division due to chromatin condensation. This is a simple process. For suppose this is a chromosome, this middle region is called as centromere. Here we have one kind of protein called as um, cytochrome, like start with K if I'm not wrong. Yeah, this is okay. Then this is chromosome become visible. So this is simply condensed like this kind of structure. Our DNA is so long, like 3.4 angstrom, so it cannot be easily fit. So what we do, we take this chromatin, this thread like structure, and add some kind of histone protein. Why? Because DNA is acidic and histone protein is basic. So then they form a new structure called as nucleosome. So this is because then this entire process is visible because of chromatin condensation. This means if chromatin is this long, it becomes condensed form only for this region. Okay, let's go to another question. What this is, when all available substrate molecule bind to enzyme, the enzyme are said to be, so when this happens, so the enzymes are called as saturated. This means no more substrate or no more enzymes are required. Which feature about DNA in prokaryotic cell is correct? This is only happen in the prokaryotic have single circular chromosome, whereas eukaryotic have symbol like not circular, but they are like linear, some different shape. Okay, the correct statement about allosteric activator and inhibitor are, so allosteric activators, see if this is enzyme, okay, and here we do get a shape like this. So this region is called as active site, this is enzyme, this is substrate. So the region, instead enzyme instead attaching to this active site, it get attached to different part of in substrate like here. So this site is called as allosteric site. Okay, so allosteric activator are same, inhibitors are simply which like stop the chemical reaction. Enzymes are simply to speed up reaction, inhibitor means to stop, inhibit something, right? So this is what statements are right. So let's see, the right both bind to region outside the active site but have opposite effect. Yeah, this is the right option. Why? Because as I mentioned, instead of binding to the active site, now they get attached to some substrate part, right? So they both bind to the region outside the active site. So this means A is the right option, none other, because only inhibitor induce. No, both bind directly to the enzyme active site. This is quite opposite, which means it's wrong. Let's go to another question, which is when does the formation of enzyme product complex occur? That's occur during the substrate convention to the product, not to absence of perfect. All these are wrong. The organelle formed by Golgi complex. So Golgi complex not formed because lysosome, peroxisome, glycosome, these all are manufactured by these. Lysosome is a different peroxisome only present in plant. Lysosome is a membrane organelle present in both, glycosome as well, right? So mitochondria is already an independent organ which can work itself. Same goes for chloroplast. Let's see another question which says primary structure difference between rough endoplasm reticulum and smooth endoplasm reticulum is RER has ribosome, SER does not. So rough, why their structure is rough? Because they do have some protein like structure on their like body, on their structure. So that's why we say RER is rough here. RER is rough because their structure have protein that's make them rough and different kind of structure, but SER does not. This is the right option. Let's go to another. The inhibitor that bind directly to enzyme active site. So we have three types: competitive, non-competitive, uncompetitive. So these are like different things. For suppose if you say competitive inhibitor, that works like simply the active site get attached. Non-competitive, where instead of attaching to this one active site, they attach to allosteric site, which I mentioned. And the last type, which is called as uncompetitive, which is also called as anti-competitive inhibitor. In this, it take place bind only to the complex but like format but not any other. So this means here the right option is C. The role does the Golgi complex play in plant cells? So plant cells forms the cell plate during cytokinesis process when cell gets divided. Okay, this in like cell structure, cell chapter, but here it forms the cell plate during cytokinesis because chlorophyll is produced by chloroplast, not any of these. Okay, conduct photosynthesis, no, that even happened during the plastic. We need some structure. Stores starch granules, no, the either is wrong. The temperature at which an enzyme work most efficient, that's called as optimum temperature. Even we say for human body we have optimum temperature which is 37 Celsius. And yeah, then let's go to another question. The permeability of the mitochondrial outer membrane is due to presence of neither cristae nor porins phospholipid bilayer. This is in plasma membrane. Cristae are the like some curve like structure present. So the permeability of mitochondrial outer membrane. Outer membrane. This means the outer membrane could be the right option. None of these here. Okay. Or maybe I think mitochondrial outer membrane due to presence of, they do have intermembrane space as well. So any of these could be right, but cristae no. Friends, some pores either not. Okay. Rest we have still so many questions remaining. Okay, let's do the inward folds of the mitochondrial inner membranes are called as, that's called as cristae. These are made to increase surface area. In Golgi topic we have some structure called as cristinae, some like NAE. So this structure is in Golgi, but cristae are for mitochondrial. So make sure you remember this. Ribosome primarily worked in genetic information processing. Ribosome because they ribozymes are different structure which were given by Sedman and Altman, like those scientists. Thomas F. They says the molecule in ribonucleic acid function is enzyme, so they are called as ribosomes. Okay, the Golgi complex is most prominent in which type of cells? That's in glandular cells. Neither all of these. At 10 Celsius increase in temperature usually happens when the reaction is double. Okay, neither stop, and these all this means B is the right option. Mitochondrial are known as the powers of cell because they make ATP. That's why this is quite important for MDCAT exams. Like this is the most repetitive exams of the MDCAT. So I hope you learned this powerhouse of cell, which is mitochondria. Why? Because they produce ATP through aerobic respiration. The models that could explain an enzyme's ability to catalyze a reaction slightly misfolded. Misfolded means you can change the shape. That's happened in Dewsfit model, not in lock and key, which was given by Emil Fisher. He just says we need a particular key, particular substrate to bind. Both models, no, it's wrong. Fluid mosaic model, no, this is about plasma membranes given by uh, John Nixon, okay, Singer and Nixon. How does Golgi complex possess protein received from RER? So they simply modify and sort them to transport. So Golgi complex work is to transport, modify all these. So I hope it's clear. Rest we'll do in the upcoming video. Thank you so much for watching. I hope you're liking this series.